So Vivet is a French-based incorporated company with a strong affiliate in Spain where we do a lot of research. And actually, the incorporation was really based on a strong collaboration and unique collaboration with what we call the CIMA Center in Pamplona. It's where the San Fermin Fiesta is taking place, for some of you may know. And it's like an applied center of translational medicine close to the University of Navarra, where the origination of the Wilson disease was started. So what we have in terms of when we incorporated the company is really to generate a company that will be focusing on liver metabolic disorders, but also understand that when you start thinking about treating such liver disorders, you're going to have to treat some patients which are pretty young. So we wanted to focus on the some disorder which are pretty unique, but also understand how we could improve the sustainability of expression, more specifically in children through our construct. So we built a pipeline of different indications, and we have a strong IP on all of this indication. And also we have strong investors, which initially are actually funded by Roche Venture, Novartis Venture, Columbus, which is a Spanish fund, and we raised 37.5 million. And in March, we announced a partnership and collaboration with Pfizer, where Pfizer has a strong collaboration with Wilson and an option to actually acquire the company. Having said that, it's important to see that the Wilson disease is really specific in a sense that it has a very long cDNA, and the main work that's been incorporated is the development of a specific construct, truncated DNA, that actually make it compatible with the size of the AAV. So the lead programs in terms of highlights of the company is a Wilson, where we'll be filing and start the clinical programs in 2020. We have a high-value program on different indication, and I will give a specific presentation on PFIC3. We have differentiating modular platform regarding the uncertainty of sustainability of expression, and that's very important for the children, either improving sustainability or finding ways to actually retreat patients or prevent the occurrence of NAB. And we have some collaboration on manufacturing to support not only VTX801, but the whole development pipeline. So what is Wilson disease? So the Wilson disease is a disorder that has actually an orphan disorder, which we have designation in Europe and US. But more importantly, it's a disorder that actually is considering almost 20,000 patients, if not more, with 12,000 being diagnosed. We all have a small protein in the liver called ATP7B, which is a copper transporter. And this lack of protein will actually, in a non-treated disorder like Wilson, lead to death, basically, at some point. The challenges were clearly over the current challenges as unmet medical need is that key points is 13 to 15% of patients will end up with liver transplantation, which can improve both, of course, liver and neurological outcome. But more importantly, you can see that this protein is really focusing on improving the ceruloplasmin production, but also transporting, more importantly, the copper in the cells with the excess copper in the cell through the bile and eliminating through the bile, basically, and not being reabsorbed. And the current treatment actually don't treat the main, I would say, consideration and treat symptoms and accumulation of the copper. So it means that we're eliminating copper through chelation. So what we see in this treatment, like dipenicinamine or trientine, is that 50% of the patients are not compliant, and clearly some of them, a third of them, will experience very severe side effects, which can have neurological consequences, which can be irreversible and are very damaging in young population because it's diagnosed around the age of, let's say, 15 and 20 years old. Last but not least, 55% of the patient will show some neurological improvement, meaning 45% will not show neurological improvement. And last but not least, a quarter of this patient will continue despite treatment to have neurological consequences or liver consequences and disease progression, and up to 50% will end up with liver transplantation. So having said that, it's important that we characterize the model and understand how we can mimic the clinical situation in a mice model as much as we can. So what we did is characterizing over time the knockout model from the inflammation, from the cell necrosis, for the hepatocyte hypertrophy, and you can see over this histological slides some very significant hepatocyte hypertrophy, lots of inflammation and infiltration. And what we did is to understand what kind of patient are we going to treat, at what stage of the disorder we can actually treat them. And if you treat early the patient, it's clear that you see that there's some significant death in the animal model, and with the treatment, we actually fully restore the lack of mortality, and you will see the other parameters. But what we want to restore clearly is the ability with this ATP7B protein to get this expression in the liver, restore the copper homeostasis, the normal way of elimination, which is a bile, and therefore we're restoring also all of the parameters, including the ceruloplasmin, which bound the copper we have as a free copper in the blood, which can be neurotoxic. And that went through the optimization of the copper mini ATP7B developed by our Chief Scientific Officer, Gloria Gonzalez in Spain. So what we did is, through this characterization, is to understand at the different stages of the disorder, by treating with the AAV construct, what does it mean in terms of copper improvement. So this is the evolution over time of the copper. There's a massive accumulation in the male and female, and when we treat at different stage of the disease, 6, 12, 16, or 20 weeks, and you remember the histological slides of the disorder, you can fully restore normal values, which are acceptable values for the copper accumulation in the liver. In the meantime, if we look at the neurological concentration and the brain concentration on the copper level, we can clearly control this level of expression. So it's important, because of the restoration of the ceruloplasmin level, we can avoid the free copper to penetrate into the brain and affect the brain structure. For example, a patient can have the age of 20 to 30 years old, they have severe tremor or Parkinson's symptoms, which are extremely significant and could be irreversible. And one interesting thing is to think about what could happen in human. And if we go back in the 70s, basically, they started to inject through IV injection copper-64. And the best way to think about, are we restoring the copper homeostasis through expression of the protein in the liver, is to look at what's happening when you inject IV of copper-64. It's like a microdosing, basically. So when it's maybe difficult to see, but if you look at the dark spots, you see in normal patient or heterozygous, they have significant elimination of the copper-64. But when you go in undiagnosed treated patients or treated patients with chelators, you don't see any copper-64 elimination. Thus, the excess copper being chelated and eliminated in through the urine. So what we did is we tried to mimic the situation, and we did that with a knockout model. And what we can see here is that basically we have the wild-type animal where you see the elimination from the liver with the PET scan through the bile and through the intestine. Same for heterozygous. But when you look at the wild-type, the Wilson disease animal, you have a strong accumulation in the liver. And 3 months after treatment, when we see the effect starting 3 weeks after, actually, you can see this elimination. And if we do an excretion mass balance over 24 hours, we can actually restore this level of elimination in the heterozygous, which have zero sign of the disorder. Accumulating this kind of information is important in a way that we can mimic the same situation in human. So what we're going to do is a relatively classical phase 1 escalation in dose ranging in male and female and different patients. But what we're going to do is clearly mimicking this copper-64 approach. So what we'll do a classical copper-64 baseline, where normally we shouldn't see any elimination in the feces. And then after 14 weeks, we get a readout of a new copper-64 analysis, and we can classify the patient as responder or non-responder. This has been validated through the FDA and EMA as a strong outcomes in terms of proof of mechanism, and we know as a biomarker, the copper homeostasis is well established. So that's something we will start in 2020 and giving us some strong insights of this ability to fully restore the level of expression in the liver. If you look at the other kind of indication, and here I'm talking a much more severe indication and more ultra-orphan targeting children, this progressive familial intrahepatic cholestasis are focusing on biliary disorders. And we have in our liver some significant important protein to avoid the bile salts to be the detergent of the hepatocytes, basically. And here we have the bile salt, which is the BSEP. It's a genetic mutation on the PFIC2. And we have another one, which is the one on the phospholipids, which needs some phosphatidylcholine content in the bile salts to avoid the detergent property by itself. And these two mechanisms are leading to accumulation in the hepatocytes, and this can lead to very severe pruritus, and it can lead to severe liver failure. And you will see that most of the patients are transported either before adolescence, or they can be transported during adolescence, or they can have a STOMI bag where you have a derivation of the bile elimination to prevent this liver accumulation. It's extremely debilitating, and it leads to some significant liver transplantation focus. Just as an example, on the PFIC2 a natural history study, you see that from the age of 10 years old, almost 50%, more than 50% of the patients will have liver transplantation, and it goes really fast. Similarly, for the PFIC3, if you look at the evolution of the disorders of the patient, this is 10 years old, more than 60% of the patient will have liver transplantation. And that's extremely significant in terms of disease impact and life, quality of life, and potentially death for patients. So what we've done a specific construct on PFIC3, which is a large gene actually, and we've optimized this construct, and we've treated early animals in the model, and you can see the level of expression we have is pretty high compared to the knockout, the wild type, and the male treated after 12 weeks. And when we look at the bile salts elimination, this is really high in the untreated, the saline animal. This is a normal wild-type animal, and this is a treated animal. And the same for the liver enzyme, so we can normalize the liver enzyme. So what we clearly looking forward is, if you look at the histology, which is extremely detrimental in terms of fibrosis, if you look at the male or the female, and this is a two example of the male patient where we can revert the fibrosis, and this is going to be published in Nature Communication in the next few weeks. Interestingly, if you just to show the size of the spleen of the saline animal versus the normal animal, and we can both normalize the spleen weight and the liver weight actually. So this is clearly transformative for the PFIC patients, and so we're going to start the IND enabling studies, and we're targeting an IND filing in 2021. And clearly, in terms of portfolio and strategy, we're going to apply to this Wilson disease or PFIC2, where we have a proof of concept established. We have a lead candidate. We're considering more information on the PFIC2. We also have Citral Anemia, which is a super ultra orphan disorder. It's 1 out of 1,000 births, basically. It's extremely low, and we have a fifth indication where we will reveal more information next year, considering that this indication is targeting children and also targeting adults, but we need to improve the sustainability of expression in this population. Otherwise, you more convenience treatment when you treat adults. So it's important that in this technology we're developing, and it's under IP, so you would apologize me for not presenting too much data, but that would clearly improve the sustainability. And when we say sustainability in gene therapy, I strongly believe that we can remove the uncertainty we have around the potential loss of expression over time, which actually gives some potential barriers for payer conversation, and also for the ability to say we have a cure at the end of the day. So we remain committed clearly to the liver metabolic disorders, and we look forward to have more information and really curing this patient for Wilson and other indications. And thanks for the Vivet team.